Hello, my name is Ray Archuleta. I'm going to talk to you about nutrient management, but from a different approach. We want to talk about nutrient management from using this type of conceptual approach called biomimicry. How to do nutrient management from emulating nature. In the past, I've talked to you about mimicking nature. It is incredibly important to understand how you approach the natural system, but it's also very critical how you look at the natural system. So I'm going to talk to you about three major conceptual points I want to talk about in nutrient management. Keep in mind, I used to look at nutrient management from a conventional agronomist view, a very prescriptive way, not a holistic way. Now I have completely changed the way I look at it as a soil scientist and agronomist. These 3 points I'm going to talk to you in this PowerPoint. One, keep in mind most of nutrient management was based on the wrong premise. We looked at nutrient management from an absolutely incorrect way because we viewed the soil incorrectly. We looked at nutrient management, at least I did, as soil as a chemistry set with a little bit of bacteria. Now I look at it as a complete, total different way. It's a living, dynamic ecosystem. The second point I want to say, all soil tests are wrong and some are useful. I'll build upon that and explain that later. And the final one is, approach is critical. The way you approach your testing is very critical, and the way you approach your understanding towards the soil is, I cannot stress, very critical to your success. So let's keep that in mind and let's start this. First thing I want to talk about, it's about context, context, context. I don't care, I have been all over the country. I have flown thousands, millions of miles, been in every state. I will tell you, I've been on thousands of farms, hundreds of thousands of farms. All of them are degraded soil ecosystems. It's very important to understand your context. If you see the picture on the left, most of our soils came from forest or prairie systems. That is a picture showing of that same soil. It used to be 6 to 8%. And with our type of farming, we have shifted that soil. Same soil, can you believe, is 1 to 2%. That is our conventional farming systems. So when I walk into an operation, I always assume a degraded context. Context is absolutely critical to understand if you're going to understand soils correctly. We are dealing with a degraded context. The thing that I learned in college was called the mineral theory. Now, this type of theory gave us a very reductionist view of the soil. What I learned in college, that if you imagine water coming in, it becomes into the soil profile, and then it goes into the soil solution, and then in my concept, I felt like the water was this magical force that brought all the nutrients out of the mineral, and we call that diffusion. And then there's mass flow, that the roots would take up all those nutrients, and then the roots would intercept those nutrients. It was like this giant chemistry set. After graduate school, soil chemistry, that's what I learned, the mineral theory. Was this wrong? Yes, it was, because it didn't give you the whole picture. Let me build on that. The mineral theory, because of this view, and it's not just my view, but many professionals have given us a misconception of how the soil really works. This is how the soil really works. Let's build on this. Because of this mineral theory, we have created a huge problem in our oceans. If you can see that picture on the left, you can see this huge basin called the Mississippi Basin, where all these nutrients are leaking and creating huge eutrophication zone. Hundreds of thousands of miles. We are losing 40 to 60% of our N and P. And this is a problem. What we have done, because of this kind of theory, we have created bare and fallow soils throughout the country. We have decoupled the carbon and nitrogen and phosphorus cycle. Because of this concept of the way we view nutrients and how nutrient cycling occurs has created a big, huge problem. A lot of our producers think this is part of the nutrient cycle. It is not. That is a chemical salt. This is a nutrient that the soil does not prefer. It is a salt. We have to be very careful how we use this tool. It can be used as a very destructive tool that breaks up organic matter and it alters pH. This is a tool that we do not want to use and get away from it, because not only does it cost huge amounts of energy, but it's degrading our resource. We have to be very, very careful how we use this tool. This is the correct model that I want you to view the soil. And it's not just my perception, but many researchers are now coming to this new model called the ecologically based model. Dr. Drinkwater from Cornell does a beautiful job explaining this ecologically based model. This is the model of life. It is this organisms breaking the residue on top, living plants. It is they who make nutrients available in the soil. The soil nutrients are coming from many, many sources. The nitrogen is. It is these organisms that, as they release these powerful enzymes and their secretions of life, they're the ones that bring the mineral out of the rock, out of the geology. This is driven by plants and microbes. This is the correct model. So does the mineral model happen? Yes, but it's this one. It's biochemistry dictates inorganic chemistry. It's the chemistry of life that dictates what happens in the soil. It is these organisms. They drive nutrient cycling. So the more we disc them, the more we till them, the more we apply fungicides, insecticides, the more we disrupt their function. They are designed to do this. They are the nutrient cycle. I've shown you a slide before. All of our soils are a mixture of sand, silts, and clays. Geology. But it is biology, the excretions of life and death of these organisms, carrying out their function. So you have geology plus biology creating these super molecules that create soil. That's what soil is, this dynamic matrix. And it is driven by this living root. I cannot stress this. How are we going to bring phosphorus out of the rock? Right here, them in synchrony with the microbes, with arbuscular mycorrhizae fungi. It is feeding these copious amounts of organisms. 1 tablespoon can have up to 7 billion bacteria, more people than on the planet. It is this that drives nutrient cycling, that brings the potassium, the sulfur, and all the minerals out of the rock. It is these organisms, this plant, with these organisms together in synchrony that bring boron, zinc, iron, and it brings it into the solution. You can see these two charts here showing that not only do they bring nutrients out of the mineral, but they can degrade incredible toxins. They can degrade Roundup, pesticides, herbicides. But here's the problem. When you overburn the natural ecosystem, you degrade their function. These tools are harmful to these organisms. We have to be incredibly careful how we use these tools. Also, many producers like Rick Clark has talked about how he builds nitrogen, that he gets nitrogen from the atmosphere, which is absolutely correct. But the nitrogen is also coming from organisms in the soil that fix nitrogen on their own, apart from the plant. Nitrogen is coming from many, many sources, and I'll talk about that in the next couple of slides. Another slide that was incredibly powerful, this research they've done in the past is showing arbuscular mycorrhizae. Those are radioisotope dots. And they're showing how organic nitrogen is being carried by the arbuscular mycorrhizae from the soil right to the mothership, the plant. Those are radioisotope dots. The funny, when that paper came out, I laughed because the Russians knew this since the 1920s. They knew this since the 1930s, that plants can take up organic nitrogen. I was taught in school, these are the only forms of nitrogen: nitrate and ammonium. We're saying that's not true. There are other forms of nitrogen the plant can take up. They can take up organic forms of nitrogen. In fact, a new study by Dr. White has shown that plants can actually suck up microbes. They're like carnivores. That's the term rhizophagy, root eating. In other words, these plants have the ability to suck up the microbes like giant vacuum cleaners, strip their outer shell, and get nutrients from them. Who would have thought that plants are carnivores? Our ecosystem is so complex. But the moment that you look at things singularly, not holistically, you're lost. The nitrogen is coming from many sources, not just from one. It's not just from legumes. It is coming from many, many sources. What we are dealing with, ladies and gentlemen, is in this incredible ecosystem. This is a 3D model showing this subaquatic ecosystem created by cottage microorganisms. Create these aggregates that look like cottage cheese. They look like a big chocolate cake. This is what we're dealing with. Those aggregates change every 27 days. It's this beautiful system is dynamic and it changes daily. Who would have thought that this incredible matrix, it's held by life, by biology. It's like a living skin. It is absolutely awesome. So the next slide I'm going to talk about again is context. Please keep in mind, it is incredible to understand ecological context. A majority of our farms came from forest or prairie. This is a picture of secondary succession. What does that mean? If humans cease to exist, if you live in Illinois or Indiana, most of it would go back into a wooded environment. If humans would cease to exist, the natural ecosystem would shift in that direction. In the prairie, it would go back into grass. Why is it important to understand this context? It is critically important because the biology shifts in every one of those is totally different in each of those stages. If you look at the far left, this is where we're at in the majority of our ag land. Annual weeds, bacteria-driven, leaky systems. We don't want to be there. We want it to be more fungal. As we go more into the shrubs, into the trees, and emulate the natural system, the more efficiency our ecosystem becomes, the more we hold nutrients, the more the soil functions. And so, we want more fungi in our ecosystem. So if you do not understand this context here, it is very hard for you to understand what's going on and how to use your tools, how to create a shift. Keep in mind, right now, we are on the far left. A majority of farms are very bacteria-driven systems. We want to be more fungal. So we want to shift it that direction. And the way we do it is put more carbon, less chemical and physical disturbance. This slide will show you the difference between cropland, prairie, and forest. Look at the bacteria count, very similar. But look what changes. Now keep in mind, this is a measurement for a table, 1 gram of soil, 1 teaspoon. In the cropland, it's only got several yards of fungi. Look in the prairie, 10 to 100 yards of actual fungi strands in that 1 little teaspoon of soil. Now look at the forest, all the way up to 40 miles long in 1 tablespoon of soil. The largest organism on the planet is a fungi. It is incredibly critical to understand the context of your soil and where it's at. This really helps. with weed control and nutrient cycling. This slide also shows that I can easily walk up into a farm or an operation and I can know where they're at. If farmers are having problems with too much weeds, it shows me they are doing way too much disturbance, not enough carbon, they're hurting the fungi, they're disking, tilling too much, they're overgrazing, bacteria dominant system. I want to shift our system to more a fungal dominant regime. That's where I want to be. But you can only do that by reducing the disturbances and adding more carbon into the system. That is critical. More carbon flow, more nutrient cycling. This is Dr. David Johnson from New Mexico State. This man has taught me quite a bit. This man got his PhD at 50 years old. He went back to graduate school at 50 years old. Got his PhD at 60. He was a builder. I would recommend that every person that wants to get their PhD, they need to go work and understand the real world before they go into academia. I have learned so much from this man. I wish he would have been my sole professor when I was at New Mexico State. Let me show you some of the new research that he has brought forth. And I wish that this would be taught everywhere. In his experiment, he shows chili plants. You see the number right there is 0.14 fungi to bacteria. If you see the numbers, as you see, it goes all the way to 5.3. What that is showing that the more fungi per bacteria, the better the plants do. So the top slide shows 0.14 fungi per bacteria, but look at the one on the bottom, the far lower right corner, 5.3 fungi to 1 bacteria. Why is that important? It is incredibly important. What his data shows, when plants are very small, the first thing the plant wants to do is to make association. It doesn't matter how much nitrogen's in the soil. It doesn't matter how much phosphorus in the soil. It doesn't even care how much potassium's in the soil, or the sacred scroll everybody looks to, organic matter. It doesn't matter to the plant. What was the most critical, according to his data, was this: the fungal to bacteria ratio. I cannot stress how incredibly important that is. What does that mean? What that plant wants, the most critical relationship it wants, it's not nitrogen, it's not phosphorus, it's not potassium, it's not even organic matter. It wants to make relationship with fungi, with life. That plant needs to make that relationship so that it can get access to the nitrogen, to the phosphorus, to the potassium, and to the organic matter. I had a lot of misconceptions about how the soil functioned. I thought it was a little chemistry set. This next slide is I'm going to show you a little video. And again, going to a biologist, as an agronomist, I'm going to shamefully tell you I did not understand that. Where does a majority of our nutrients come from the soil? Let's listen to this biologist explain where a majority of our nutrients come from.
You look down at the little seedling sticking up out of the dirt. It's hard to imagine that one day this small tuft of green might grow to be a tree that towers over you. But where will this tree get all of the materials it needs to grow? From the soil? From the water? What about from the air? Like humans, plants are made of mostly water. But when you take away the water, where does the rest of the plant's mass come from? If you remove the water from our bodies, you would find that carbon makes up most of the rest of our mass, or weight. The same is true for plants. Humans and animals get carbon from the food they eat, which is often plants. So where do you think the plants get carbon? They get it from the air. The air around you seems empty, but it's not. Air is made of tiny bits called molecules. When you look closer at the molecules in our air, you'll find that it's mostly nitrogen, oxygen, and carbon dioxide. Plants actually absorb this carbon dioxide from the air through small openings in their leaves or other tissues. These openings are called stomata. Once inside a plant, the carbon dioxide is broken down, and some of the carbon ends up in the chloroplasts. Chloroplasts are special cell parts that hold chlorophyll, the molecules that make most plants green. Chloroplasts are also where photosynthesis takes place, which is how plants convert sunlight energy into food they can use. Carbon, water, and energy are used in this process to make glucose, which is a type of sugar. Glucose molecules can then combine to form long chains called cellulose, which are used to build plant structures like cell walls. So the carbon from the air, along with water and energy from the sun, help to make these structures. As cells grow, they divide and make new cells. These new cells are how plants get bigger. The most amazing part of this process is changing air into plant material. This sort of magic trick transforms something which seems weightless and empty into material that has weight and that you can see. From a tiny seedling to a tall tree making seeds of its own, plants grow by pulling carbon out of the air. So in the end, plants really are made out of thin air.
Let me tell you, I did not understand that. One of the things that we get criticized all the time from other agronomists or other people in the agriculture field, well, if you go down this regenerative movement, you're going to suck all the nutrients out of the soil. We've just shown you that a majority of the nutrients, 95% of the plant's biomass, comes from thin air. It comes from hydrogen, oxygen, nitrogen, and carbon. When you're out there walking and you're seeing that incredible cornfield, 95% of it is from the air. Well, I used to think it was from from the soil nutrients. And so when you think of it from a chemistry mass balance, first things agronomists tell you, you're going to mine the soil. You're sucking all the nutrients. I took a picture of the redwood trees. My wife is in the background and my daughter. That tree weighs 4.2 million pounds. 3.99 million pounds is from the air. And I'm going, wow, that was an incredible epiphany. Parts per million of phosphorus and all the other nutrients come from the soil. A very small amount of the nutrients actually come from the soil. You have to understand that. It's from biology, and very little bit of the nutrients come from the soil. Now, please don't misunderstand. Can you mine the soil? Absolutely. You can outstrip the biogeochemical nutrient cycle. You have to be careful how you do farming. But if we do it right, it can be perpetual. Now, here's the second part I was going to talk to you about. This was a very profound statement by Dr. Buzz Clute, good friend of mine. He says all soil tests are wrong, some are useful. He got that statement like from another person who said all models are wrong, some are useful. We have to be very, very careful when we use our models to mimic the natural ecosystem. They are incredibly deficient. So what do I mean that all soil tests are wrong, some are useful? There is no one single soil test that will ever tell you everything that's going on in the soil. It's like when a person goes to a medical doctor. A person is an incredibly complex ecosystem. That's why a good doctor will use all kinds of tests. A blood sample, urine, fecal, CAT scan. You use all the tools to show indicators of function. Soil's the same way. It is incredibly complex. I use many of the soil tests. I don't use just one. But if you don't understand and have a basic understanding of soil function and what is going on, all the tests are useless to you. You have to have a basic understanding of ecology and the soil ecology and how it works. Here's the thing. It's the way we farm that is destroying nutrient cycling. Notice how I call tillage. Tillicide. Side means to kill. Tillage. overhaying, over application of pesticides, too much fertilizer, they diminish the function of the soil organisms. So I have a basic concept model that I use, or an acronym, to help me realize to be very careful. This was taken by Dr. Stephen Gleesman in an agroecology book. The acronym is FIST. Why do I like that? Why do I like it? Because what do most of us want to do in nature? We want to force it and control it. What I'm showing you here is you have to be very careful of the frequency of your disturbances, your applications of your chemistry, your tillage. Frequency means a lot. It really is important how frequent is your disturbance. The intensity of your disturbance. Did you vertical till or did you plow? Plowing is incredibly disruptive. The scale of your disturbance. Did you just spray one part of the field or did you spray the whole farm? If you spray insecticide in one field that you were having the problem, it's quite different when you spray the whole farm. Then when the neighbors hear that you sprayed, then they go spray the rest of the neighbors. Then you diminish the functionality and the self-healing, self-organizing, self-regulating mechanisms that control the pests. It is incredible the scale of your disturbance, and the timing of your disturbance is critical. So frequency, intensity, scale, and timing. Why am I telling you about this? It's about this. So if you are continually using chronic stress, and let me explain the difference between acute stress and chronic stress. If you go eat fast food once in a while, your body can handle it. That's an acute stress. But if you keep eating poor food and not exercising, that is chronic stress. If you drink copious amounts of alcohol, eat poorly, you stress the body. It's called chronic stress. It'll collapse and you'll get sick. The ecosystem's the same way. Chronic stress, too much tillage, too much fungicide, you hurt the Arbiscum mycorrhizae, then you hurt the springtails, then you hurt the rest of these organisms, and the system collapses. Then you have to go buy more chemical inputs. So if you disrupt the functionality of these incredible soil web, then you have to spend much more money on inputs. Let me show you the little video from Michael Thompson, a farmer, talking about is showing no-till, cover crops, and grazing. Look at the life of those organisms. This is nutrient cycling. Who would have thought these organisms are critical to the water cycle? Without these organisms, you cannot create aggregates and keep pore space open. Now look at this high input no-till. A lot of chemistry, but they're doing no-till. They're applying one principle, but look at hardly any life. Let's go back and show you again. Look at the difference in this slide. No-till covers grazing, life. Look at this one. Hardly any life. It does make a difference what you're doing. It matters how you manage the living plant. Again, context. A lot of times we get a lot of blowback with the new soil test, the Haney test. People say, well, Ray, it's not calibrated. And I'll respond back to the researchers, either is your test. Your test was calibrated in destroyed soil ecosystems. You didn't even understand the context. You didn't even understand that the soil was alive. The current soil test is very inadequate. I use that test just for very few parameters. You have to be very careful with that old soil test. That old soil test was built on the wrong premise. To build my point even more, there's a great there's a great YouTube video by Dr. Brad Joran. He, in his presentation, he says, there must be magic at the border. His study has shown, if you look at all the states, this is a 2-year fertilizer cost based on 180 bushel corn and 60 bushel soybean. This is 2-year cost. Values are $1,000 per $1,000 per $1,000 per 1,000 acre. Notice the difference in costs. Anywhere from Missouri being $260 to, and you go across the border to Illinois, it's 300 and some. Notice the difference between our fertility recommendations. And people wonder why our farmers are going broke. I keep saying our farmers are the poorest millionaires I know. They spend copious amount of money on fertilizer. Our old tests were not accurate because they were based on the wrong premise. We've known since the 1930s so that when you put chemical fertilizer by the Magruder plots in Oklahoma, that over 60% of the fertilizer never reaches the plant. 40% of the fertilizer only reaches the plant. What happened to the other 60%? Leached and taken up by the microbes. Here we have, and we've known this for years, but it's rarely talked about. In a study done in Illinois, the Illinois Fertilizer and Chemical Association, they went and did test plots all over Illinois. And it's interesting to see some of their data, which was taken off the website, by the way. And it's interesting to show. If you see this graph, it's called the yield response curve. And on the axis on the left, you can see yield and bushels to acre, and then the bottom is nitrogen, pounds per acre of actual N. It's interesting when you show this to producers and to a lot of the agronomists, what do they focus on? They see that little triangle and goes, oh yeah, look at the optimum. 125, 130 units of N for 150 bushel corn. But they failed to see this. How come I almost got 100 bushel corn with zero fertility? Where did the nitrogen come from? Zero. I almost got it came from the soil. But what we focus on, again, looking at the wrong parameter of success, yield. Yield and profitability do not equate. It's not the same thing. Here's another of one of the plots they showed. Look at where it shows the zero plot. What is this graph showing me? This graph is useless. Unless a farmer puts a zero check on your operation and do a half rate check, full rate check, your information is worthless. You need to know where's the fertilizer coming, where's the nutrients coming from? Is it from the soil or is it from the fertilizer? Unless you put a zero check, your data, your information is worthless. It is incredibly important to realize that. A lot of farmers get upset. I said, look, if you are not doing your own check strips, and doing your own research, you are going to be subservient to everybody selling you product. Keep in mind, you do your own research. Here's another graph showing Dr. Rick Haney put all that data together. And what this graph shows out of those 170 plots, notice those dots represent one of those plots. In the zero, you can see it, and from the zero fertilizer, it ranged anywhere from 50 all the way to 160 bushel corn. And if you look to the right, the optimum level was about 150 units of N. And yet, as you keep putting more fertilizer, the efficiency went down. It got worse. So we have been applying way too much fertilizer for many, many years. This graph shows you the efficiency. In fact, the less fertilizer you apply, nitrogen fertilizer, the more efficient it is. The more you put out, the less efficient it becomes. So this slide also shows you, keep in mind of your context. That soil on the left, even though those were the same soils, function total different. The one on the left is nitrate dependent. Very huge amounts of nitrate in it. That is the predominant species of nitrogen in it. And the soil on the left, it has different forms of nitrogen. Now it has dissolved organic nitrogen in it. Amazing. Now you have access to a different pool of nitrogen. Now, we're going to talk about the Haney test versus the regular test. I'm going to tell you, the Haney test and the old soil test are apples and oranges. Do not compare them. One is based on the premise of life and biology. The other one is based on the premise of chemistry. Different tests, they tell you different things. I use both of them, but the one that I make my predominant decision is the Haney test. It is more accurate for nitrogen, phosphorus, and potassium. Why? It's based on the concepts of biomimicry. Awesome. We finally have a scientist say, hey, why don't we mimic nature in her approach, in her chemistry? Why don't we use water as the major extractant and root exudates? Whoa, what does the microbes and the plants see? Water. What do the microbes see? Root exudates, those gentle acids. Brilliant. Uses very sophisticated machinery. Again, this slide shows the difference of our old approach, man-made chemistry, this caustic acids, forcing the phosphorus and the potassium off the exchange side, versus Rick's approach on the right, using the gentle acids of what exudes from the roots. Keep in mind, plants giving off these gentle acids help also make nutrients dissolve. So their plants are leaking these acids, but biology is also making it more available, multidimensional. Here's the sophisticated water analyzers. Notice Rick doesn't use soil analyzers, he uses water analyzers. Why? The reason the Haney uses that is because the microbes swim in water. When it rains, the microbes swim in those films of water. I call the Haney test the most powerful passive test. It's like the blood sample of the soil. So that's why doctors love using blood sample. When you go in there, it tells you a lot about hormonal function, your pH. It tells you where you're at. It is a great passive test. Snapshot, tell you where you're at. The sugars, where you at. Haney test does that. It is a single shot in time. It is a snapshot. Please understand that. Your body changes like this. The moment you eat a pizza, the moment you eat pizza, you do a blood sample before you ate the pizza, and after, it's going to be totally different. Soil's the same way. It changes daily. The old soil test only could pick up nitrate or ammonium. Haney test picks up all those other molecules in the organic form, amino acids. That's why the farmer can give way more credit for nitrogen, because we get it in the organic form. I could never understand when I did a soil test on the forest or the prairie, it would always be low in nitrogen. And I would say, now hold on a second. How come the trees are so big? Who's giving nitrogen to the grass? My soil tests are not telling me. The Haney test can pick up this difference. This is a Coffee County, Tennessee. This is farmers who had been no-till for over 50 years. Look at no-till by itself on that left picture. Once we started using multi-species cover crops, the Haney test was able to show a 279% change in respiration less than 3 years. A 39% increase in cycling of water-soluble carbon. If you trip in those fields and trip by mistake, they will consume you, the microbes. That's how quick they're cycling. A 50% increase in soil health score in less than 3 years by using covers. It is incredible, but we would have never been able to pick up that difference without the Rick Haney test. Also, the Haney test has the ability to pick up all those molecules that the biology on the top of the surface break down. And those rains bring those complex carbon molecules that feed that system. You can see where you start off with a carbon ratio of 80 to 1. That's 80 carbon molecules to 1 of nitrogen. Notice as it gets decomposed more, all soils will go back 11 to 1 to 12 to 1 molecules of carbon for every 1 molecule of nitrogen. What you see is that the Haney test will show any from a range of 6 to 1 to 20 to 1. Once we go over 20 to 1, you start shutting down nutrient cycling because there's not enough nitrogen in the system. The Haney test has the ability to help you see that. Again, we're able to measure those incredible molecules. This is kind of an imagery showing, think of yourself in a microbe, and here these molecules are floating into the water. in the water, they're in the water column. Microbes are going there, stripping those carbon molecules, eating, consuming. This is the food. This is water-soluble carbon. A lot of people brag about organic matter. Folks, it is not the organic matter, it's the water-soluble carbon. In other words, you can have a big house, 30% organic matter. I've seen soils with 30% organic matter, and yet there's no cycling and no plants growing in it. It's the water-soluble carbon that's critical. Look at these molecules. This is where the party is at. Here's where all the nutrient cycling is occurring. Again, the organic matter is the house. Who cares if you live in a 10,000 square foot home, but if you're eating potted meat and Vienna sausages every day? Life sucks. Can't get any work out of you. Big deal, you live in a big house. Impressive. 30% organic matter. That's not where this is at. Is organic matter important? Yes. But it's not the driver for nutrient cycling. It's water-soluble carbon. Why do they call it water? Pretty explanatory. Water, the driving force. Without water, there is no function, no cycling, period. We've been missing 50% of the nitrogen. For the last 60, 70 years, we have been over-applying easily by 50%. We have not been giving farmers credit for the organic forms of nitrogen. The Haney test deals with all these parameters. So when you take the test, it measures water extractable organic carbon, the food. The percent MAC is the efficiency of the microbes to eat the food. Water extractable organic nitrogen, because if you can't have nitrogen, nitrogen is what the microbes build their body with. The water extractable organic carbon is the groceries. They need both. You cannot separate those two. Also measures inorganic N and P, the organic carbon and nitrogen ratio, and it also measures Solvita one-day respiration. The Russians knew back in the 1900s that the soils respire every day. So the higher the respiration, that means the higher nutrients in the soil, the more groceries. So this test is very sensitive to CO2 respiration. These are the parameters. Now, Dr. Buzz Clute, brilliant scientist, did a crowdfunding with several farmers in South Carolina. What they did is, here's one of his producers, Mr. Coleman. You see here, the only thing that separates these two farms is that dirt road. This is South Carolina, very humid, very sandy soils, like sugar sand. Mr. Coleman has been doing 15 species cover crop, rolling the cover. He has not applied P and K and fertilizer and lime in years. He brought his organic matter up from 1.2 to 1.9. You're probably saying, well, that's really low. They're sand, sugar sand soils. Look at the soil on the right. 3 species, cotton and corn and soybean. Prescriptive fertilizer and lime, extractive degradative. Look at the two soil structure. What you see on the left is Mr. Coleman's. Look at the neighbor. What's the difference? Carbon. It's life. This graph does a really good job showing by Dr. Clute when each of those lines represent his 5 or 6 farmers. Look what they started with in these sandy soils. From a 0.5% organic matter all the way to 2%. Look at the organic matter after 3 years in these sandy soils, upward trend. Every one of them went upward. Look at the phosphorus went up on all of the farms. You can see how they started. Each of those lines represent one of the farms. That red line represents the threshold where they went above that. Keep in mind, after 29 months, no phosphorus added, it went up 15 pounds of P increased in the soil. None added in these sandy soils. How did that happen? I'll explain it in the next slide. Look at the soil pH. Look where they started, from a range from 5.9 to 6.2. In less than 29 months, no lime added, it went up 0.4 of a unit. Do you know how hard it is to raise pH? It is incredibly difficult to raise pH. How did these plants, these cover crops, increase phosphorus, none added, and change the pH? Pretty simple, life. Those covers went down deeply, brought the phosphorus, all those nutrients back to the soil surface, brought the calciums. It's biology, life together. Now, we're going to move on. We're going to talk about something that a lot of things that we're doing incorrectly, and one of the most detrimental things, if not done properly, is the over-application of manure in our cropping systems. I'm talking about manure coming from CAFO operations, hog operations, cattle, swine, this industrial model. We have to be extremely careful with this type of manure. This type of manure is very toxic. Please keep in mind, natural ecosystems did not store manure in anaerobic conditions. Manure was taken care of very quickly in aerobic conditions. Buffalo did not carry an animal waste plant behind them and disc the manure in. Microorganisms took care of it very quickly. Let me show you very quickly what happens in this case study done in Pennsylvania. In the green column is Jim Harbaugh. He's a dairy, and he applies his dairy slurry 3 times a year, and he only puts 6,000 gallons, and he does multi-species cover crops. He's no-till. He's been doing it for years. The other dairy puts slugs of dairy manure at 20 to 60,000 gallons per spring and fall application, continuous corn, no cover crops. If you look at that column, look at the amount of water extractable carbon. Jim's has 410, the other dairy 315. But here's what I want you to focus. Notice the respiration. Where they had covers and reduced amounts of manure, he had a high respiration rate. That means the microbes are functioning. The higher the respiration, the better cycling. Look at the one when they put all the manure on one slug, respiration was really low. But if you go down to the further column, how much nutrients was available? On the Jim Harbaugh dairy, where he splits the application and the cover crops, 270 pounds of N was available. Where the other producer just dumped it out there, shut the microbes down, only 32 pounds was available to the crop. That manure is toxic. We have to be very careful of where we get our manure. Look at the soil health score, 28 versus 7. 0 to 7 is poor, 7 to 14 is fair. Jim's was excellent. Again, careful with, this is a lot of the research papers showing we have to be very careful with our manures. Here's a research paper that I want everybody to read. It's called The Potassium Paradox, Implications for Soil Fertility. Please read that. I don't have time to go over that. But this shows by Dr. Mulvaney that a majority of the potassium we're putting out there is not helping yields at all, but actually, if you use potassium chloride, you're actually hurting yields. Biology makes it available. This Haney test will show you, this is Dave Brandt's using multi-species cover crops. Look at the respiration, 146. This is the same soil of his uncle. 40 years heavy tillage, monoculture. Look at the respiration, 12 parts per million. Look at the difference in soil health scores, 18 versus 4.4. Makes a huge difference in what you're doing out there. Here's one of my favorite tests that I use. It's called the Slake Test. People can tell me how good of a farmer. All I got to do is drop your soil into a cylinder. If it falls apart, it's telling me you're doing some things wrong on your operation. On the left, you'll see 30 years of cover crop, 40 years no-till. Look at the soil health scores by David Brandt, 18, excellent. If you see the other one, the conventional system, look at the soil health score, 4.4, a 320% change. The Haney test was able to show that. The Cornell test, another soil test, does not have the ability to be that sensitive. It is not a test that I promote. If you look at the same soils, that same healthy soil, the Cornell said it was excellent, but it called the very degraded soil a medium soil and only showed a 34% change difference between the two soils. I've been in those two fields. The soil in the right is extremely degraded. It is in the poor category. So all soil tests are not the same. So what do I do as an agronomist? I use all of these tests. I use the earth fort test, the phospholipid fatty acid test. I use the Haney test. I use the traditional test. I do tissue tests. I do stock tests. I do check strips, water sensors, the Slake Test, infiltration rings. I use the shovel. I use all of them. What kind of agronomist do you want? An agronomist that gives you a prescription, or one that looks at your whole system holistically and targets so that he can wean you off your prescription drugs? You want somebody that understands the system. We are going broke, and it's affecting our health. What kind of doctor do you want? What we teach a lot of our clients is to do their own research. You heard Rick Clark talk. Rick is a farmer scientist. He doesn't take anybody's word. Don't take my word, no one. He does his own research. This is a great guide to get you down this path. It's written by a PhD professor that wrote it for a fifth grader like me. Simple. So you can do your own research. I definitely recommend it. You can find it online, and it's a guide on farm demonstration research. Again, Soil Health Academy, Understanding Ag, these are some of our benchmark protocol. When I go consult, these are all the parameters I look at. I look at the Haney test. I no longer go to the Ward Lab. I go to Regen Ag Lab. I use the PLFA. I use these other soil parameters. Water infiltration, aggregate stability, plant bricks. Some of you heard Rick say, listen. Plants, animals, insects, birds, those are all indicators. Don't you dare have anybody tell you that your anecdotal information is not important. Every bit of information is important. And one of the most powerful things we do when we go out there, I check for skin, I check for the crust in the soil, I smell it. My most powerful tool, the shovel. Photo points. I'm going to leave you here with a couple of people that have changed, that absolutely changed nutrient management. Russell Hidrick, young farmer from North Carolina. This young man is a great manager for growing corn. He won the yield contest, but let me explain here. This young man grew up in town. Started as a fireman, got into farming. He didn't have all these filters. He watched us on YouTube. We don't get the credit. We're just messengers. He didn't have, and his local district conservationist, he had the right people influence him. He was open-minded. Let me show you what he's done. In those very degraded North Carolina soils, those Piedmont soils, they're not Indiana soils. They're not deep prairie soils. Here's the average yield that this young man has attained on his own in the last 6 or 7 years of farming. 199 bushel to 220 bushel. The county average is 112. This young man won the dryland contest in North Carolina. 318 bushel corn with less than 140 units of N using the Haney test, no-till covers. He is a great manager. So look at his fixed variable costs. Makes a huge impact on how you look at the system. Another one is a great person, a good friend of mine, is Jack Stahl. He is from the Hitherite community up in Canada. Jack went to go listen to Gabe Brown. and went to a workshop, and he was on the path to go buy a fertilizer plant. He was going to buy a fertilizer plant. And he went to go hear Gabe Brown speak. Keep in mind, he was driving over there, and he went to this workshop first and heard Gabe Brown speak. And the way the story goes, Jack Stahl is a very direct man. And he was when Gabe was telling the story, he was very nervous on the front row, and he kept moving around. Later, he finds out that Jack Stahl was trying to buy a fertilizer plant, but what Jack did instead, he listened and believed Gabe and changed his whole operation. That community, as you can see, those beautiful homes, they farm together. That is a 20,000 acre farm. In 2018, his fertilizer costs were $1.5 million. They dropped it down to $127,000. Instead of buying that fertilizer plant and cutting all their inputs, they went and bought 10,000 more acres and paid cash for it. It made a huge difference in their lives. It changed the community. He now is a believer of mimicking God's creation, biomimicry. Leaving here with the last couple of slides, when I do the Haney test, I only go to one lab right now that I trust. It's called Regen Ag Lab. Why do I only trust this one lab? Because he does it according to Dr. Rick Haney from ARS. Rick designed the test. There are many that are saying they're doing the Haney test, but they are not following the protocol. It is incredibly critical to understand that. A lot of things people say, well, the Haney test, some people have used it in California, they say, well, it's all over the board. Of course it's all over the board. If I sampled your blood at one day and do it the other day, it's going to change. The Haney test is a test, one shot gives you information for that day only, but we use it to predict our nitrogen and our nutrients, what it's going to be. That's why when we do this test, we do it when the soil temperature is 55 degrees. It is incredibly critical. You need to understand the test is very sensitive. It is not like the old test. It changes. It is incredibly dynamic. The soil changes every day. You need to understand it. A lot of problems where a lot of researchers and a lot of agronomists have with this test is because they think of the soil as a chemistry set and a static pool of nutrients. It is not. It is just as alive as you. So when I take a blood sample, that blood sample is only good for that day that blood sample was taken. Please understand that. Wrapping it up with the last 3 points. A lot of our soil tests were based on the wrong premise. That's what we knew. They were not based on the premise of life. If you do not have the right approach, the wisdom that you approach the natural system is wrong. I have seen I've seen a lot of intelligent people leave the university. But intelligence does not give you wisdom. Wisdom is this, the right goal. Mimicking nature is wise. It's got design. It's elegant. It's regenerative. Follow the design. If your science is not based on biomimicry, it is wrong. And the same thing in the medical field. That's why when they tell you and they give you a prescription and you have side effects, what you are seeing is incredible side effects of health through a lot of our natural ecosystems. That's why we're having more cancer, more disease, more health issues. It is critical how you approach it. All soil tests are wrong. Some are useful. Keep in mind, the Haney test is useful. Approach is everything, ladies and gentlemen. If you approach nature incorrectly, she will beat you with a stick. Approach her with her biology. View her from her view, from her eyes. I want to thank you again, and I'm honored to be part of this AgEmerge slides and PowerPoints and talks. Thank you.